Hi everybody, this is Mr. Matthew here with a video on DNA replication. So in this video, what we're going to talk about are the different enzymes involved with DNA replication, how it proceeds with its leading and lagging strand. We'll then talk about the nature of the semi-conservative nature of DNA replication and how you get two identical copies from an original strand. That is going to be key to knowing why DNA replication is the molecular mechanism of inheritance. So let's get to it. So here is the basic process of DNA replication involved. So we have a lot going on in this particular image, and I'm going to talk about the major components of it. So first up, let's acknowledge that we have a DNA strand here that's being opened up by an enzyme called DNA helicase, or in this case, just helicase. And what you can see is helicase is going and it's opening up and wedging the double-stranded DNA into two separate single strands. Now, this is at one end of what is known as a replication bubble, and the one side is called a fork. And if we were to go down to the other side over on the right-hand side of this picture and continue out, we would find another replication fork and another helicase opening the DNA up there. I'll show you a replication bubble in a few slides. Now, once that DNA is opened up, we are going to have these single-stranded DNA strands that we're going to be able to make copies of. So one of the key things to know, though, about DNA replication is the enzyme that drives it, known as DNA polymerase, can only bind to a double-stranded structure. So we have an enzyme called Primase, which is up here shown on the top strand, which is going to lay down a short what is known as RNA primer, complementary to the complementary to the DNA strand that's going to allow a landing spot for the DNA polymerase. The DNA polymerase will then bind there and then continuously add new complementary bases that are complementary to the template that is there. So in this particular example, you can see that there is a Primase that has laid down a DNA primer. And then what we can see is that complementary to the DNA, shown by the black strand here, opposite of that from a G, we see a C, and complement to a T, we see an A, and that's going to continue on to make complementary bases. So the DNA polymerase, as it does so, is going to be bringing in those complements, A's with T's, C's with G's, and it's going to have some directionality to it as well. And that is sort of the most complicated aspect of DNA replication. So what we'll see here is down on the bottom, you can see that the DNA polymerase is building towards the replication fork. It is going where we can only add DNA bases on the 3' end. So we say DNA builds in the 5' to 3' direction, where new bases are going to be added on the 3' end of the new strand that's being built. And DNA is anti-parallel, so it is going to be the 3' end, importantly, of that new strand being built, and not the 3' end of the template strand. Very important to note that about these components. Now, as it's building, you can see this leading strand on the bottom is moving towards the helicase, and the helicase is going to continue to open up as it unwinds the DNA. So we call this a continuous replication because we're going to lead, and it's going to lead into the fork, and as the helicase moves down, it's just going to keep moving and moving and moving and moving until this replication bubble fuses with another replication bubble. On the top strand, it has a separate issue. So it can, again, only be building in that 5' to 3' direction, and the helicase is on the 5' end. So what we can see here is that the DNA is going to be built in the 5' to 3' end until it runs into the next fragment. And so it's going to be a series of short fragments called the lagging strand fragments, and these fragments specifically are known as Okazaki fragments. And so they're going to build a series of these, and as the helicase opens up more, a new Okazaki fragment will be then added on as it has moved on. So we can see our RNA primer here is starting a new Okazaki fragment. As the helicase moves down to the left, a new Primase will start a new fragment, and they will backfill in. The enzyme that we see here, ligase or DNA ligase, is going to help repair the strand so that those fragments are actually stitched together when all is said and done. So when you're done and you're looking at this DNA, you won't be able to know which was made in a leading strand and which would be a lagging strand. You're just going to see two identical strands of DNA, or two identical copies of DNA, one on the top, one on the bottom, and you wouldn't know which was built in a leading form and which was built in a lagging form. All right, so DNA replication replicates relates to the transmission and conservation of the genetic code. And one of the interesting things I always find about this is that when the structure of DNA was first published by Watson and Crick in 1953, they actually have this line in their paper as almost like a throwaway line, that it has not escaped our notice that the specific pairing we've postulated immediately suggests a possible copying mechanism for genetic material. Full details of the structure, including conditions assumed in building it together with a set of coordinates of atoms, will be published elsewhere. So they just were like, yeah, we've realized that we've set up this situation where you have DNA, it's complementary to another DNA, and so it's pretty obvious how it's going to replicate, but they didn't actually work it out. They just sort of had this throwaway line in there. And it wasn't until a few years later when Messelson and Stahl, so this is 5 years later, we have two scientists known as Messelson and Stahl, published a paper called Replication of DNA in E. coli. And what they found out is they had three hypotheses about how this could work. They said, if I have this original strand of DNA and it gets copied, we could do it a couple different ways. One thing we could do is we can make it like a photocopy. So we have our original strand of DNA and we're going to make a copy of it. And we're not going to lose any of that original DNA. It's just going to stay as it is. And we're going to make like a photocopy and make a new version that is completely new DNA. That's known as conservative replication. Another model would be if we took that original DNA and we opened it up, and each one of those original strands serves as a template, and we make complementary copies to it. And then there was a third one, which was like, well, some sort of blend of it, so that each of them is, each strand that we have is some sort of blend of old and new, and they referred to this as dispersive. And it was in that published 1958 article that Messelson and Stahl argued and showed data to support that in fact it is semi-conservative, that the model we showed in the previous slide is in fact what happens, where each of the template strands serves, or each of the original strands of DNA serves as a template for the new DNA. So we refer to that as semi-conservative replication. So how does this structure lend itself to it? As we've just mentioned, we have this original DNA and we'll have a series of replication forks that open up, and helicases will push those out, and DNA polymerase will bind and make some complementary copies. And so what we end up seeing as a result of that on the bottom here is that the blue dot structures are new DNA and the solid black line represents the original DNA. So in the copies, they are half new and half old, and this is referred to as semi-conservative.